Hiya! Today we're going to be discussing the biochemistry of auxins. So just some history quickly. The first one was discovered in the 1930s, and this was indole 3-acetic acid, or IAA, with the structure shown here with the indole ring and an acetic acid joined to it. And the definition of an auxin is the ability to stimulate elongation in stems, coleoptile, and rooting. Auxin has absolutely loads of biological functions, so I just listed a few of them here, like shade avoidance. This is where you get rapid elongation of the stem at the expense of like your leaves, your storage organs, and you also have accelerated flowering. And this requires rapid Auxin synthesis. Gravitropism, so in the root you have a myeloplast. These sense gravity and cause a change in expression of PIN, which is an Auxin transporter, which we'll see later. And you transport auxins to the lower side of the root where it inhibits root cell elongation and therefore promotes growing downwards. Fruit formation, also fruit ripening and fruit abscission as well, and finally leaf abscission. Moving on to structures, first looking at IAA, we see it's got an indole ring and a carboxylic acid group. Now the distance between the carboxylic acid group and the aromatic group in auxins needs to be about 0.55 angstroms, which will not be conserved across my diagrams. Here we have IBA, which has a longer chain of carboxylic acid group. You can also add substituents to the indole ring, like in 4-chloroindole 3-acetic acid. And instead of an indole ring, you can also just have a phenol ring, like PAA shown here. Looking at them all together now, indole acetic acid, IAA, was the first one discovered and is the most common throughout plants. However, IBA actually induces rooting more effectively than IAA, and IBA and IAA interconvert. 4-chloroindole acid is very common in legumes, and phenylacetic acid, PAA, is used for interactions with the rhizosphere. There are two main ways to make auxins, the tryptophan-dependent pathway, discussed here, and the tryptophan-independent pathway. Now, starting with the dependent pathway, we have the central metabolites PEP and erythrose 4-phosphate. These are central metabolism intermediates, and they're used in the Shikimate pathway to create charismic acid. And also, the Shikimate pathway is used to create folates and other aromatic amino acids. Now, charismic acid or charismate is used in the chloroplast to create tryptophan. This is through a series of reactions, including intermediates such as indole 3-glycerol phosphate and an indole ring. The last enzyme is tryptophan synthase beta. So there are actually 4 ways to get from tryptophan to indole acetic acid. I'm just dealing with the most common one, which is through indole pyruvate. However, another example is through tryptamine, and this is only seen in brassicaceae. So looking at the enzymes, first of all, we use a transaminase, which is pyridoxal phosphate dependent, to get indole pyruvate, and then we need a monooxygenase, which uses NADPH and oxygen to get from indole pyruvate to indole acetic acid, which uses a decarboxylating mechanism. As mentioned previously, there's also a tryptophan-independent pathway of making auxins, because the tryptophan-dependent pathway cannot explain all the auxin accumulation that can be measured in vivo. In the vegetative state, this tryp-independent pathway is actually the dominant one, and the mechanism for synthesis is from indole and indole 3-glycerol phosphate. Auxin activity can be controlled by conjugation. These conjugates can be amides, glycosyl esters, or larger molecular weight species like glycans. Examples of amino acids used in amides, you can have aspartate, glutamate, glycine, or the valine shown here. Examples of sugars that can be used, you've got glucose shown, you've got myo-inositol, and you've got things like rhamnose. And they can be degraded in the case of aspartate or glutamate conjugates, or temporarily stored. Another example of storage or possible storage isn't necessarily conjugation but interconversion, shown at the bottom here, because indole acetic acid can be interconverted with indole butyric acid, using a similar reactions to what you see in fatty acid elongation and degradation. Auxins such as indole acetic acid, IAA, can be inactivated by oxidation. This is using reactive oxygen species, or ROS. Now these ROS can be generated through NADPH oxidases, which are actually stimulated by auxin addition, creating like a negative feedback loop. And these ROS are actually repressed as well by flavonoid species. Now on to moving the auxin around the plant. Most auxins are actually produced in the shoot apex and young plant leaves at the top, and must be transported down to the lower organs. This transport can be either passively in the phloem or more actively in polar transport, which is a cell-to-cell transport occurring in the vascular cambrium, and its derivatives that are partly differentiated. So polar transport requires transport across cells, and so we need to get it into the cell and then back out of the cell. So focusing on getting it into the cell from the outside to the inside, we're looking in the apoplast with a pH of 5.5. The pKa of indole acetic acid is about 4.75, so we've got a good distribution of both the ionized and the non-ionized species. The non-dissociated indole acetic acid can cross through the membrane. However, the ionized form must pass into the cell through a transporter, and these are lax transporters. In the cell, however, the cytosol has a pH of 7.4, shifting the equilibrium towards to favour the dissociated form of the auxin, and therefore cannot pass through the membrane passively to be removed from the cell. It must go through specific transporters, and these are called PIN transporters or PGPs. PGPs are phosphoglycoproteins, and they are very important in long-distance auxin transport. Focusing now on the more characterised PIN efflux transporters, these can be used often to build auxin maxima, which can be used as a developmental trigger, for example, in specifying sulfates. There's 8 PIN transporters in Arabidopsis. They are expressed throughout the plant. They're often positioned with polarity in the cell membrane, which means that they can be specific directional transport of auxins out of the cell. They can be found in the plasma membrane and in the ER membrane, and are actually required to mediate all effects of auxins. And each subtype has specific effects often associated with it. For example, PIN1 in organogenesis versus PIN2 for kind of tropisms. The current hypothesis about how PIN transporters are put on specific parts of the plasma membrane is through phosphorylation. This phosphorylation can occur through membrane-bound AGC kinases and can be reversed by PP2A on this large intracellular loop labelled in red here. However, the mechanism by which this works is not actually known currently. Our first auxin receptor, tier 1. So just to set the scene here, SCF E3 ubiquitin ligase complexes, what these do is they bind to specific proteins in the cell, and they then tag them with several ubiquitin molecules. This then targets them to the proteasome for degradation, and so removes them from the cell. Now, how they target it is using F-box proteins, which often has some substrate specificity, for example, binding to a phosphorylated protein, for example. Here, the F-box protein is tier 1, our auxin receptor, and with no auxins, what they do is they can't bind to AUX/IAA, the green molecule here, and this allows the AUX-IAA to go unhindered, and its main role is an inhibitory protein of these ARF proteins, auxin response factors, which are transcription factors. So without auxins, you've got an inactive E3 ubiquitin ligase complex, and you've got an inhibition of transcription by these ARFs. When we add auxin to the system, this yellow circle, we allow the ring-shaped tier 1 protein to bound to these inhibitory AUX/IAA proteins, which allows them to be polyubiquitinated and then degraded. This degradation of the inhibitory protein leads to the activation of these ARF transcription factors, these ARF transcription factors, which can then bind to the auxin response element on DNA and control the transcription of several genes. Some of them that are upregulated include the SAW proteins. These are small auxin up RNA, that's the kind of acronym, and these are highly unstable proteins, some of which have been seen to be involved in cell elongation. There's also GH3 proteins, which are involved in conjugation of auxins to amino acids, like we discussed previously, and the AUX/IAA proteins, which are inhibitory, these green proteins shown here. Now, what's not included in this diagram is all the different isoforms of these different proteins that are involved, and how you can have specific combinations of like your TER proteins and your AUX/IAA proteins, and these create different affinities for different auxins. You also have different kinds of ARFs, which can then bind to different AUX/IAA proteins and create different inhibitory heterodimers. This creates a lot of cell specificity and specificity of response. The second receptor to discuss is SCIP2A. This is an F-box protein, just like before with tier 1. And what it does is, without auxin, it can't bind to transcription factors this time. These transcription factors are E2FC and DPB, which could bind to DNA and prevent the G1S cell cycle transition, and so reduce the proliferation rate of cells. However, When we add auxin, just like before, they can bind to the skip2A protein, allowing these transcription factors to bind to skip2A, allowing them to be polyubiquitinated and then degraded. And so auxins will then promote this G1S transition. The final auxin receptor to be discussed is APB1, which was actually the first discovered. However, not very much is known about it. So it's commonly found in two places, in the Ariad lumen, where there's pH of 7, and in the apoplast, where there's a more acidic pH. Now, if you assay the affinity for auxins of APB1 in these two different pHs, you find that there's much higher affinity for auxin in the apoplast pH of about 5.5, and a very, very low affinity for auxin at pH 7. And so therefore, it's thought that APB1 is actually only functional as an auxin receptor in the apoplast. However, it doesn't have any transmembrane segments, and so to commit its information across into the cell, it's thought to require some transmembrane containing proteins. One of these proteins is thought to be SKU5, and is thought to be some other proteins as well that help this GPI-anchored protein SKU5 commit its information into the cell. Once the information is into the cell, it's thought to cause ion channel modulation, and is also thought to determine clathrin-mediated endocytosis of PIN transporters, these efflux transporters we talked about before. This is through some ROP proteins, which are Rho family GTPases. Much like cytokinins, auxins crosstalk with quite a lot of other hormones in plants. For example, with cytokinin in the root meristem. So together they control the size of the root meristem, both pathways converging at the SHI2 gene. Cytokinin favors differentiation, whilst auxin favors increased meristem size, so they're actually antagonistic here. SHI2 is actually activated by cytokinin signaling and degraded by high auxin by the SCF-TIR1 complex, which we discussed as the first auxin receptor. Now, SHI2, when it's active, i.e. with high cytokinin signaling and low auxin signaling, reduces PIN1, 3, and 7 expression and reduces cytokinin synthesis through regulating IPT genes. So moving on to ethylene now, at the bottom, we're looking at root hair elongation. And in root hair elongation, both auxin and ethylene are positive regulators. They act synergistically here. However, when we start looking at root hair initiation, again, they're positive regulators. However, this time, ethylene's effect, instead of being synergistic with auxins, with them signaling pathways converging, we have ethylene actually increasing the amount of auxin biosynthesis. So just to summarize everything that's been said so far, first of all, I'm going to start on the outside of the cell. So we've got Auxin transport by these PIN1 efflux transporters and import by these LAX or AUX transporters. And you've also got APB1 controlling ion channel modulation and the clathrin-mediated endocytosis. Moving into the nucleus, we see that the levels of activity of these SCF ubiquitin ligase complexes are dependent on the concentration of auxins, with high auxins activating these E3 ubiquitin ligase complexes and therefore promoting degradation. Now, they degrade either these green AUX/IAA inhibitory proteins, or they degrade these transcription factors E2FC and DPB. And so therefore, when there's low auxins, i.e. low levels of degradation, you only have transcriptional control by these E2FC DPB proteins, which repress that G1S transition. And in high auxins, you only get transcriptional regulation by these ARFs, which bind to auxin response elements and control the expression of proteins like the GH3 genes for conjugation or those Sauer genes. Yep, that's just about it for auxins. There's a lot more in the reviews that I've put in the description, and hopefully the presentation and also this table will be useful. So yeah, next time we'll be doing ethylene.